Welcome back everyone. I am Saptaparni from BioLive. Hope all of you are doing very well. So in today's video we are going to discuss about genetic crosses in Drosophila, and today we will be focusing on triple lethality regarding to Carli mutation in Drosophila. Okay. So in this series I have already made 5 videos. In my first 3 videos we have discussed about eye color mutation, vestigial wing mutation and Carli mutation in Drosophila and related problems. And in my 4th and 5th video we have discussed about 50% and 100% male lethality in Drosophila regarding Carli mutation. You have to remember that the 100% male lethality is considered as double lethality in Drosophila, whereas when there will be another lethality added to it, then that type of lethality is known as triple lethality. What is that addition? We will learn in a bit. Okay. So without further delay, let's begin, and I would like to request you to kindly watch at least 4th and 5th video of this series before watching this one. Okay, then the video will be very easily understandable to you. So before beginning, I would like to request you to kindly subscribe to my channel for more such videos, and don't forget to hit the like button if you really like this video and share it with your friends. Okay. So first of all, what is Carli mutation? See here, the Carli mutant allele, which is denoted as CY, is phenotypically dominant over the wild type wing allele denoted as CY plus. So you can see here the Carli mutant allele is dominant over CY plus. So we know that the allele which is dominant shows its phenotype in both homozygous and heterozygous condition. So similarly you can see here in heterozygous condition CY plus is showing Carli wing phenotype in Drosophila. But interestingly you can also see here that in homozygous condition the CY is not showing Carli mutation, rather it is showing lethality. So now the question comes, why this is showing lethality instead of Carli wing phenotype? Because you have to remember that the Carli mutant allele is not only the dominant allele over CY plus, it is also a recessive lethal allele. So that is why in homozygous condition CY shows lethality. This progeny does not survive. Okay. And obviously as CY plus is recessive to the CY, so obviously in homozygous condition CY plus will show wild type wing in Drosophila. Now this is the wild type wing in Drosophila, okay, Drosophila fly, and when the mutant allele is present the wing becomes curled backwardly, which is known as Carli mutation or Carli mutant phenotype in Drosophila. Okay, got it. Now coming to triple lethality. In my previous video, I have discussed about what is double lethality. Double lethality means when there is 100% male progeny is absent. Okay, so for that we have put two lethal alleles L1 and L2 on the X chromosome. Okay, and you have to remember that these lethal alleles L1 and L2, okay, the L1 plus or the wild type of lethal allele is dominant over its mutant type, that is L1, and similarly L2 plus is dominant over its mutant type allele, that is L2. Okay, if L1 is present then no lethality occurs, if L2 is present no lethality occurs, but if L1 L2 is present then lethality occurs. Okay, so I have discussed about all this in my previous video. Please kindly watch that video too. Okay, then it will be very easily understandable to you. Now this was double lethality. Now to introduce the triple lethality or the third lethality, we have to introduce L3. Now what is L3 and where should we introduce it? See here the question that is given here. You can see here that there is only, this is the information regarding F1 progeny data. Okay, and here you can see no male progeny is present here, so obviously the double lethality is occurring here. 100% male progeny is absent. Along with that you can also see no wild females are also present here. Okay, so no wild females are also present here, only Carli females are present, which is a mutant phenotype. Okay, so to kill all these wild females you have to introduce a third lethality, that is the lethal allele L3, okay, lethal allele L3 to kill all the wild type progenies or all the wild type females here. So where should we put the L3 allele, lethal allele? So see here, as it is killing the wild type females, okay, so we have to put the L3 allele on the autosome carrying CY plus or the wild type allele of the wing phenotype. Okay, so to kill the wild type individuals, we have to introduce L3 or third lethal allele on the autosome carrying CY plus or wild type allele for wing. Okay. So now see how to derive the P generation cross for this problem. Okay. So see here, as there is no male, so we have to introduce the double lethality on the X chromosome of female. Okay. Why? See here, the double lethal alleles are L1 and L2, and here in the female you can see one X chromosome is carrying L1 plus and L2, whereas another X chromosome is carrying L1 L2 plus. Okay, so here you can see they are carrying the lethal alleles in complementary type. Okay, that means this female is this mother is surviving. Why? Because though here L2 is absent, but its complementary L2 plus is present here. Though here L2 is present, but its complementary allele L2 plus is present here. Though here L1 is present, which does not produce any protein that is produced by L1 plus. Okay, but here due to presence of L1 plus, it is complementing the work of L1 here. Okay, so that is why by the process of complementation, the product that is needed from L1 plus and L2 plus are present in female, so female is surviving. And in males you can see the only X chromosome is carrying both the lethal alleles in wild type condition, so that their product is available and male could be viable. If they carried any one allele as mutant allele like L1 and L2 plus or L1 plus L2 or both the allele in mutant from L1 L2, then they could not survive. So that is why male have to carry both the lethal alleles in its wild type condition. Okay, so that is how you can introduce 100% male lethality. Why? Because when these X chromosome will combine with this Y chromosome, okay, it will not get the product of L2, so it will die. Okay. Similarly, when this X chromosome will combine with this Y chromosome, it will not get the product of L1. Okay, so it will die. So that is how you can introduce 100% male lethality in this question. Okay, no male should be present or should be viable here. Okay, now to kill all the wild females too, okay, so we have to introduce the third lethality known as L3. Now see here, we have introduced the lethal allele L3, mutant lethal allele L3, specifically on that chromosome which carries CY plus or wild type of allele for wing structure. Okay, so here in this condition we have taken CY plus for mother and father both, okay, in heterozygous condition, and the L3 mutant allele is exclusively present on the CY plus carrying autosome. Okay, so that when these will combine with this, okay, this X chromosome will combine with this, no one X chromosome from these two will give L3 plus product, so it will get dead. Okay, so it will be lethal too. Okay, got it. So one question may arise in your mind, why I have put an I here? Because let me first delete all these things, then I can tell you. So see here, the L1 and L2 are present on the same chromosome and they have a tendency to recombine. Okay, so to stop this recombination between L1 and L2 loci, we have put the inhibitor of recombination here, so that L1 plus L2 plus and L1 L2 could not be formed by the process of recombination. Okay, and if this forms, then 100% male could not be dead here. Okay, so that is why to stop the recombination we have put an I here. To know more about this, please refer to my previous video. The link is given in the description box below. Okay, where I have discussed about double lethality or 100% male lethality. Okay, got it. So let's do the cross here and let's see how many gametes could be formed from these two parents. Okay, see here. So see here, from mother, four types of gametes could be formed. One is CYL3 plus, L1 plus L2. Okay, then CYL3 plus with L1 L2 plus, CYL3 plus with L1 L2 plus, then CY plus L3 with L1 plus L2, CY plus L3 L1 plus L2, then CY plus L3 L1 L2 plus, CY plus L3 L1 L2 plus. Okay, from father to we can get four types of gamete. One will be CYL3 plus L1 plus L2, CYL3 plus L1 plus L2, then CYL3 plus and Y chromosome. Okay, then CY plus L3 and L1 plus L2 plus. Okay, then CY plus L3 and Y chromosome, CY plus L3 and Y chromosome. Okay, so these 8 types of gametes are formed, and now let's do the checkerboard for the F1 generation progeny. Okay, so these are the F1 generation progenies here. From the checkerboard you can see we have put the male gametes over here and female gametes over here, and here you can see these are the 16 progenies that could be formed from this cross. Okay, now see how many of them will survive and how many of them will be lethal from their genotype. Okay, so see here, it is a female, you can see, as it has 2 X chromosome, but as it carries CY which is recessive lethal allele, so it is a lethal female. Okay, next, here also the same condition, as it is carrying 2 CY alleles, so it is lethal female. Okay, lethal, and as it is carrying 2 X chromosome, it is a lethal female. Now next you can see here, this is carrying CY plus, so it is Carly in phenotype and it has 2 X chromosomes, so it is female. And now why it is not lethal, why it is surviving? Because see here, the wild type of alleles of L1, L2 and L3 are present. Okay, so the lethality could not occur here. Next, the similar thing is here, CY plus, so it is a Carly. It has 2 X chromosomes, so it is a female. Then it has wild type L3 allele, wild type L1 allele, L2 allele, so it is surviving. Okay, so these two are surviving females, Carly females. Then next, see here, CY, so as it has CY, so obviously it is lethal, and as it has X and Y chromosome, it is a male lethal. Okay. The next, see here, as it has CY, so it is lethal, and due to presence of X and Y, it is a male progeny that could not survive. Then CY plus CY, so whenever there is CY plus CY, it should be a Carly one, but you can see here, due to absence of L2 plus, this progeny could not survive. Okay, though there is L1 plus, L3 plus, but there is no L2 plus, so it is lethal male. Okay. Next here, due to absence of L1 plus, this male progeny could not survive, so it is also lethal here. Then see here, it is CY plus CY, so it is Carly, 2 X chromosome female, and it has L1 plus, L2 plus and L3 plus alleles, all alleles in wild type conditions, so it is a surviving female, Carly female. Okay. Similarly, this is also a surviving Carly female. Due to CY plus CY, it is Carly. Due to 2 X chromosome, it is female, and due to presence of L1, L2 and L3 wild type alleles, it is a surviving female. Okay. Then see here, this wild type female CY plus is not surviving. Why? Because you can see here, though there is presence of L1 plus and L2 plus allele, but there is no presence of L3 plus allele. Okay, that is the thing we have tried to make by putting L3 on the wild type allele carrying autosome. Okay, so that when these two will combine, no L3 plus can be derived from this. Okay, so though it has L1 plus, L2 plus, but due to lack of L3 plus, it is lethal female. Okay, got it? It is very important why it got dead. And here also similarly, due to absence of L3 plus, it is a lethal female, which is if it survived, it could be of wild type in nature, but due to absence of L3 plus, this female got dead. Okay. Then next, see here, This male is dead due to absence of L2 plus. Okay, so it is lethal. This male is dead due to absence of L1 plus, so it is lethal. Then this male is dead due to absence of L2 plus. It is if it survived, it could be a wild male, but due to absence of L2 plus, it is dead. Then this male is dead due to absence of L1 plus. Okay, so it is also dead. So you can see here, out of 16 progenies, okay, only 4 curly females are surviving. What are those 4 curly females? If I let me first remove all these marks from here so that you can see very clearly what are those 4 curly females from here. Okay. I would request you to do the cross by yourself so that you can be very clear about what is going on here. Okay. So see here. These are the 4 curly females. These are the box numbers I have mentioned here for your reference: 3rd, 4th, box number 9 and 10. So these are the only 4 curly females, box 3, 4, 9, 10, which survived. Okay, and other 12 progenies got dead. Okay, now what are those progenies? Among those 12 dead progenies, which you can see there are obviously 4 females survived, so 4 female will be dead. Among those 4 females, 2 females are dead due to presence of CYCY, recessive lethal allele. Okay, so see here, due to presence of CYCY from box 1 and 2, these 2 females are dead. Okay, box 1 and 2, and 2 wild females are dead due to absence of L3 plus. We have seen here the box 11 and 12, you can see these 2 wild females due to CYCY plus, but these are dead due to absence of L3 plus allele. Okay, that we have introduced here as the 3rd lethal allele. Okay, so these 2 are dead, 11, 12, box number 11, 12, due to absence of L3 plus. So that is the calculation about females. Now let's get through the 100% male lethality. So all of these are males. Okay, and 100% males means 8 males could be survived from here, but all are dead. See here the cause of their death: 2 males are dead due to CYCYs, the recessive lethal allele. Those are present in box 5 and 6. See here in box 5 and 6, you can see here these 2 males are dead due to presence of CYCY, recessive lethal alleles. Okay, next see here, 2 curly males are dead due to absence of L1 plus, box number 8 and 14. See here the box number 8, you can see here this male is dead due to absence of L1 plus allele, and here box number 14, you can see this curly, if this survived, it could be a curly male. So okay, so this curly male is dead due to absence of L1 plus allele. Then one wild male is dead due to absence of L1 plus allele in the box 16. So this is the box 16. Here you can see if it survived, it could be CYCY plus, that means a wild male, but it is dead due to absence of L1 plus allele. Okay, got it? And also for the absence of L3 plus allele, you can also say that. Okay, here 2 curly males are dead due to absence of L2 plus allele in box 7 and 13. Let's see here, where is box 7 and 13? Here you can see as it has CYCY plus, if it survived, it could have curly phenotype, but it is dead due to absence of L2 plus allele. And in box 13, let's see here also the same thing, if it survived, it could be a curly male, but it is dead due to absence of L2 plus allele. Okay, and next, 1 wild male is dead due to absence of L2 plus allele in box number 15. Okay, so see here the box number 15, you can see here if it survived, the male could be a wild male, but it is dead due to absence of L2 plus allele, and along with that absence of L3 plus allele too. Okay, in the box number 15. Okay, so this is the reason behind the death of total 12 progenies. Okay, and the survival capability of 4 curly females. Okay, so I hope and from this you can see that only curly females survived. So that matches with our question, only curly female survived, so our cross is completely correct. Okay, so if you have any doubt till now, you can leave a comment below in the comment section. I will try to answer that. Okay, and if you like this video, don't forget to hit the like button and share it with your friends, and don't forget to subscribe to my channel for more such videos. Okay, thanks for watching and happy learning.